Hi everyone, thanks the organizers for giving me the opportunity to present. Today I'm going to present scJoint, transfer learning for data integration of at a scale single-cell RNA-seq and single-cell ATAC-seq data. Why do we need to integrate single-cell ATAC-seq with single-cell RNA-seq? First of all, single-cell RNA-seq and single-cell ATAC-seq characterize the cells from different aspects. Integrating two methods will allow us to have a better description of the cell identity. Secondly, due to the sparsity of single-cell ATAC-seq data, it does not always have enough power for cell type identification. And lastly, since single-cell RNA-seq technology is relatively mature and widely used, there are a large amount of public single-cell RNA-seq data with well-annotated cell types that can be used as reference to transfer labels to single-cell ATAC-seq. There are several limitations of the existing integration methods. For example, these methods are only tested on simple scenarios, and they are very sensitive to the feature that is selected. Also, most of these methods require large memory and also long computational time. To address these challenges, we have developed an end-to-end training method with joint update between dimension reduction and label transfer, named scJoint. The input of our method includes gene activity scores matrix from single-cell ATAC-seq and gene expression and cell type labels from single-cell RNA-seq. The method consists of three main steps. The first step is a joint neural network-based dimension reduction and a semi-supervised transfer learning. This is based on a neural network that consists of a joint embedding layer and a prediction layer. The output from the first step is a joint embedding of the cells from the single-cell RNA-seq and single-cell ATAC-seq. Then, in the second step, we transfer the labels using k-nearest neighbors on the joint embedding we derive from the first step. And after we have the transfer labels, in the third step, we will rerun the model with an addition loss function to obtain a better joint virtualization and refine the transfer labels. Next, I will show three case studies of scJoint, and the first one is the mouse cell atlas, and the second one is the integration of multimodal data, and finally is the analysis of a pair data. First, mouse cell atlas. We apply our method on two mouse atlas data from single-cell RNA-seq and single-cell ATAC-seq. The mouse cell atlas of single-cell RNA-seq is tabular from 20 organs and tissues generated by two technologies, SmartSeq2 and TX Genomics, with 73 cell types in total. The ATAC-seq data are sequenced from 13 tissues with 81,000 cells and 29 cell types. Overall, there are 19 overlapping common cell types between the two datasets. Here we show that in the results of the overlap cell type integration. The first row is colored by the cell types and the second row is colored by the technology. We can see that our algorithm is able to remove not only the batch effect within single-cell RNA-seq, but also the modality effect between single-cell ATAC-seq and single-cell RNA-seq. At the same time, scJoint will prefer the cell type signals, and we achieve a more than 10% higher accuracy rate compared to other two methods. This observation can be confirmed by the evaluation matrix. Here we use zero coefficient to evaluate how good the joint embedding of different methods in preserving the cell type signals and batch effect removal. On the axis here, a higher coefficient indicates the cells of the same cell types are staying closer to each other, while higher values on y-axis here indicates how well the batch are mixed. Therefore, the best method should be on the top right corner, which is scJoint, as it achieves the best performance in balancing the modality effect removal and cell type signal preservation. We also perform a stability analysis on the atlas data by downsampling the cells from single-cell RNA-seq as training data. Compared to thread, scJoint maintains a high and stable performance even when only 20% of the cells from single-cell RNA-seq datasets are used. This suggests that scJoint can be applied in practice where only a subset of single-cell RNA-seq data are annotated. This leads us to our first take-home message today. scJoint shows robust performance on highly heterogeneous atlas data. And next, we will show scJoint's ability to incorporate additional modality information in the integration of single-cell RNA-seq and single-cell ATAC-seq across different conditions. To illustrate this, we perform an integrated analysis of scJoint and aseptic data from PBMC. scJoint is one of the recent multimodal technology that sequence gene expression and measure protein abundance in the stem cell. Similar to scJoint, aseptic publishing this year is able to sequence open chromatin and protein abundance in the stem cell. Here we use a PBMC data from two conditions. One is control and the other one is stimulation that with CD3 and CD28 activated. Overall, scJoint generated a better joint virtualization of two technology. Especially for subtypes of T cells, they are clearly separate from each other, and also from the t-SNE, we can see that the two technologies are well mixed. Moreover, scJoint is the only method that able to reveal the difference between the two conditions. We can see that compared to B cells and neutral killer cells, the naive CD4 plus T cells from two conditions are clearly different from each other. Again, looking at the transfer accuracy rate, scJoint outperforms than the other two methods by a large margin, where we have 87% accuracy rate, significantly greater than thread and corners. This leads to our second take-home message today. scJoint enables accurate integration of single-cell multi-omics data. Finally, I will use RNA-seq data as gold standard to evaluate the performance of scJoint. We compare scJoint with two other methods that designed for paired data. We can see that in the joint embedding of scJoint, the cell types are clearly grouped separately. And looking at the evaluation metrics, we found that scJoint outperformed than the other two methods. scJoint is also very computational efficient. We use scJoint to integrate millions of cells from human fetal atlas generated by RNAseq and a single-cell ATAC-seq. Due to memory limitation, most of the methods are not able to handle the data with more than a million of cells. We found that scJoint is the only method that can integrate millions of cells in a short period of time and also achieve a high accuracy rate. This leads us to our last take-home message. scJoint is computationally efficient and consistently achieves high accuracy rate. In conclusion, our method is able to integrate at a scale single-cell RNA-seq and single-cell ATAC-seq data from heterogeneous tissues. And scJoint is able to construct joint embedding for two modalities that remove batch effects and also preserve the cell type signals. And lastly, scJoint is able to transfer labels from single-cell RNA-seq to single-cell ATAC-seq data with high classification accuracy rate compared to existing methods. Lastly, I would like to acknowledge this project is with close collaboration with Tongyu and Shen from Wimond's group in Stanford. I would also like to thank my two supervisors, Rachel and Jing, and all the members from Sydney Precision Bioinformatics Research Alliance. If you are interested in more details of scJoint, especially the methods detail, you can check our manuscript in bioRxiv and also try our method, which is available in GitHub. Thank you.